can show you a screen now.
Yeah. We are waiting for a few more students to join in.
Yes, we'll start.
We'll start.
One minute. I'll give you a brief introduction. Good afternoon to all. On behalf of Chairman Director of Principal Staff and Students of the Upstream College of Engineering, I'm Salma, Assistant Professor at Department of Biotechnology. Being a pleasure and privilege in welcoming our alumni, Mr. Abdul Wahid, for today's annual webinar series on Upstream Process Development, Best Practices, Scaleup and Technology Transfer. So, most of you all know Dr. and today's remember he is our first batch part-time student from 2003 to 2007. He started off with he did his MTech in KIT University, Bangalore, and he started his career in Biotechnology Limited as scientist. So he's been through quite a few upgraded posts like senior scientist, principal scientist, and currently is in the position of MSAT Associate Manager with 10 plus years of experience and our management and department update expert very well known that and now I request our HOD, Dr. Vijay Margana, to say a few words about today's series.
Welcome, Abdul. How are you? I'm fine. How are you, sir? I am fine, thanks. Indeed, it's a pleasure to invite you to the department whenever we do some kind of activities, and you are kind enough to accept it whenever we invite you, and we are thankful to you for that. And dear students, we are proud to introduce our first batch alumni, Mr. Abdul, and I want to see some prefix in your name, Abdul, as a doctor or something like that, so that we can you can make us some more proud by adding some prefix to your name. And
Yes.
students, you have your senior with you now today, and you can freely interact with him, whatever doubts you have, even in the case of academia or higher studies, or in joining the companies, or in framing your career. Whatever doubts you have in your mind, you can freely ask your senior. And we always invite our alumni to interact with you people. And whenever we conduct any such activities, Abdul is always available to us, and we are really glad and thanks to Abdul. And Abdul, you can interact with our your students and your juniors, and you can motivate them to take up the career as you are taken up. And without wasting much time, I request you to take over the session and start your session, Abdul.
Yeah. Thank you, sir. Thank you for the introduction. Yeah, it's always been a pleasure whenever I'm invited because it brings back all the memories of having gone through the BTech biotech education. So nice to be here. So for all the students, you can already see my mail ID there. So if you haven't noted down, you can always note it down right now, because usually I do this at end of the session, but the slide has been up for some time, so you can take this. So as Salma ma'am told that I am right now the person responsible for the manufacturing sciences upstream team. So as part of this function, what I'm doing is I'm doing the technology transfer for some of the biosimilar molecules at Biocon. So we have built Asia's largest biologics plant at Biocon, and then the kind of whenever we talk about technology transfer, right, what are the things which are more important, or what are the things which we have gone through in the past, and how did we overcome those challenges? So all this, the presentation is about this. So if you have any doubts, I think the instructions were already passed on, that you can just put it up, or might be we can take all the questions together once. So because I'm at Biocon and today I'm at work, so you can see me wearing the mask, right? So during this difficult times, we have adopted to virtually conduct all our businesses. But then, life science is a place where without you being there in the lab, nothing moves. So that is why you can see most of Biocon right now is working at 100% capacity. So what is the mission, vision, and what we are? So Biocon is a integrated biotech enterprise of global distinction. So when I say integrated, what it means is right from cloning to a vial of, say, the cell bank, to the final vial in the market with Biocon brand. That's what we are aiming to be, and not only a partner with other companies. So and the vision is to enhance the global healthcare. So this is not only by innovation, but also bringing in the affordable biopharmaceuticals to for patients. So we have our partners, the healthcare systems, and all this which help in achieving our goal and the vision. And whenever I say Biocon, right? So Biocon is a group of companies. So right now, the biosimilar business has been hived off as Biocon Biologics, which I am an employee of. So Biocon also works on the small molecules, the novel biologics, which is again Biocon Limited and Bicara, and branded formulations and research services. So research services is majorly the Syngene arm of it, and there is also a contract manufacturing part of the uh, Biocon group of companies, which is also comes under Syngene. To take you through the timeline, so as all of you might know that in 1978, it's been more than 40 years now that Biocon is in existence. So we started off as an enzyme company, and in 2004 we were listed as a IPO, and around this time, which is 2004, we decided to go ahead and transform into a complete biopharma company, thereby selling all the enzyme business. And around 2007, this is the interesting time. Around 2006 and later, this is where we entered into the manufacturing of novel biologics. In fact, Biocon was the first company to launch a novel biologic, which was the Biomab EGFR for head and neck cancer. And around 2009 to 2015, so this is the phase where I joined Biocon. It was around 2010. So we had R&D partnerships with Mylan and other companies, and the result of this was in 2000, you would have come across, right? This is something which is out in the public domain. We got approval for trastuzumab biosimilar drug in 2017 December, and we are marketing the product over there and also Europe. So that tells a lot about company, because usually what happens is in biologics is a complex business. So having someone take up the business and also go ahead and market the product with all the approvals, and there have been so many audits till date, and all the audits have gone without any 483s, and we have been able to like continuously supply the market with the drugs manufactured at Biocon. So we will be going through some of the case studies, which were the practical challenges which we had faced through in the scaling up of the bioprocesses, and also as part of the learning, what are the considerations for a successful scale up and technology transfer. So cell culture scale up. So whenever you talk about bioprocess scale up, right, the most difficult part always lies with upstream and not downstream, if you see, because we work with living cells, and living cells have a mind of their own. So you have to keep them happy. So how do you keep them happy depends on how good you are at maintaining the cell culture process parameters. So as part of routine scale up, usually the process which is developed at a smaller scale bioreactor, which is visible right here. Okay. So are you able to see the pointer? Salma ma'am or Manjuna sir, can you confirm if you can see the pointer? Just a second. So the 1 liter bioreactor here. So the task is getting from here to a pilot scale bioreactor like this, which is usually the volume ranges from around 50 liter to all the way till around a few hundred liters to a bigger scale bioreactor. So if you have read about some of the bigger companies like Samsung Biologics, which is in South Korea, or some companies in US, the scale is around 15,000 liters for mammalian cell culture. So what do we do in cell culture scale up? So the first and foremost priority being, because the scale up means that the volume dependent parameters have to be professionally scaled up. But at the same time, you have to also manage the volume independent parameters at the same set points, because unless you do that, then you'll not be able to do a proper scale up. And whatever volume dependent parameters are there, these always cannot be scaled up proportionally because there are a lot of parameters which goes in. So if you have come across this, then you are subject of mass transfer, where you have the theory of mass transfer coefficients, right? KLA. That brings in that is in picture. And also, irrespective of the scale or configuration, the objective is to provide the microenvironment of the cells to be similar from a small scale all the way till the production scale. Why? Because if the cells are in the same environment, they will produce the same kind of a product. So we might think, looking at all this nutrient waste, metabolites, and also the process parameters, that it is very complex. In real, if you do understand how the mammalian cell culture works, then it is not so complicated. So there is a lot of variation which can happen in the microenvironment of cells. For example, when you are sparging in CO2 to maintain a pH in a carbonic acid based buffering system, you could see that there are localized concentrations which could be causing for the pH to be lower. Or for example, if you are going with a very high agitation, you could see that the shear is higher. So all these things can greatly influence the way you get the titer or the product output from a batch, to all the way till what kind of post translation modifications happen to the product. And apart from these two, you also have the temperature and agitation. So because bigger the vessel, you'll have challenges in maintaining the temperature, because cell culture, the reaction is exothermic, but it's not exothermic to the extent where it can become a hindrance for the cooling solutions. But in microbial, usually this is the case where it is highly exothermic and you have tough time maintaining the temperature in the fermenter. And see, once we understand that what needs to be controlled, for example, the pH, DO, CO2, and nutrients, then you know like how to control it. So one thing could be that you have this flowcharts over here, right? So you do know that for pH, you need to devise a pH control strategy. That is, either you have to devise a CO2 addition or CO2 stripping. Same goes for oxygen, that you can always decide how much sparge ratios of air versus oxygen needs to be used. And also for temperature control, we have already discussed this. For pressure control strategy, you know that as the pressure increases, the dissolution of gases also increases. So as you go to larger scales, You'll also have issues with having a very different concentration. Although the DO probe would read, say, for example, 40% or 50%, it might not actually be 40 or 50%. It could be, say, 60 or 70%. It all depends on the pressure. So in order to you have the microenvironment or the cells to be the same, you have to manage all these pathways before you even decide on going ahead with any optimizations. So during the development, what would have happened is that the R&D team establishes that what is the kind of temperature the cells grow. For example, most of the cells, right, the Chinese hamster ovary cells or the NS0 cell line. So these non-secreting mouse myeloma cells, that is, or BHK cells. So all these are the type of mammalian cells which are used commercially for production of monoclonal antibodies, which is the business we are in. So what R&D does is they determine the temperature. So for example, the Chinese hamster ovary cells, right, they have an optimal of around 36.5 degrees. So if you know that 36.5 degrees is something which is optimal, then you obviously know that if you go to higher temperature, then the cells might not grow. And if you go to lower temperature, the same might be visible. So there needs to be a ranges for the temperature as well as pH and DO for the cells to grow. So these become your scale independent parameters. And the bioreactor at scale, say for example, say a 10,000 liter or a 15,000 liter, you have to characterize the bioreactor in terms of, say, what is the P by V. When we say P by V, this is something like the power per unit volume. So if you have gone through all the textbooks which are there in the engineering college, then you can recall that power per unit volume is nothing but the total volume exerted by the impellers onto the liquid. So the power per unit volume is dictated by the individual power number of the impellers. For example, the Rushton has a power number of 5, the marine impeller has a power number of 0.35. We have newer impellers which could have a power number of say 2 or 3. It depends on which manufacturer you are getting it from. And based on the P by V, you can decide to do the KLA studies. That is exactly the gassing strategy. So we had talked about this, that you can always proportionally increase the scale dependent parameters. So for the gassing, you can devise the strategy and measure what the KLA is looking like. So for example, say at a certain vessel volume per minute, which the short form is VVM, how does the KLA look like? So what combination of the gas flow rate and the power per unit volume, that is agitator speed, can give you what kind of KLA. So as far as you're able to get this, then you can always create a fine balance so that you are not sparging excessively, so that there is too much foaming, and at the same time you are not having a very high agitation rate, which could cause the cell shear. And next is the mixing time. So the mixing time should be reasonably lower. That is that say for example, if the critical mixing time is 20 minutes, that is the time it takes for the nutrient, from the time the nutrient has been added for it to reach a cell. If that is minimized, it is good, but it can't be going to a very low mixing time as well, because that would require a very high agitation rate. Therefore, that might exert more shear to the cells. So you need to have a fine balance of the P by V, the KLA, and the mixing time. So if you understand all this, how the bioreactor has been characterized, then what could happen is that when you are actually running the batch, all this is done in a media which is mock media. So mock media could be something which has a surfactant and all the solutes, but devoid of any other nutrients or cells. So this is where all the studies happen. And once you take it to the actual culture where the cells are thrown into the picture and you have nutrients, so you need to have a optimized design for the same. So if you find out that through your computational fluid dynamics that you can achieve the required KLA in zones and there are no stagnant zones and the shear is not very high, in example, if you have a Rushton turbine, then the shear will be the highest at the point where the Rushton turbine is there. So that shear should not be the shear equal to where the cells can break. or cells can die. So these things can have to be kept in mind. And during the operations, you have to take care that what volume are you going to operate. For example, if because fed batch, you always start with a volume, and then eventually there is a lot of feeds which get added and the volume increases over time. Therefore, introducing a dilution for some processes even up to 50%. So what that means is, say for example, if you start with a volume which is just above the second impeller, which is the middle impeller, and then it touches the top volume, so that should also reflect in a change in the RPM, because now you have a higher volume, although a additional impeller has been engaged, but you need to still maintain the same P by V in order to deliver a similar KLA. Similarly, like what time is the oxygen gets used, that is also critical, because the faster the oxygen gets used, higher will be the lactate concentration. And that can drive the pH down, thereby affecting the growth rate of the cells. So we'll go through some case studies. So this is for one of our first processes where we had attempted a first time scale up of a biosimilar program. So for those of you who don't know what biosimilar is, for example, trastuzumab is a biosimilar, but the originator product is, or the innovator product is called as Herceptin. So Herceptin, it has certain quality attributes like glycosylation, charge variants, or certain impurities or non-glycosylated heavy chain. So all these other type of impurities or purity profile of a reference product. So if you are able to match it, then only it is called as the biosimilar program. So the challenges was that we don't have access to the innovators' reactor designs. We don't have access to their media or the cell lines, but we were tasked with getting the same product quality at end of the day from the production bioreactor. So what we have to start with always is the facility fit and the scale up assessment. So for example, if you have the lab scale bioreactors and we have a pilot scale bioreactor which is around 50 to 100 liters, if you have optimized the parameters here and you have been successful in scale up, you will already generate knowledge saying which are the parameters which I need to look into. And based on the data generated from the pilot scale, you can always project and say that, okay, this is the kind of KLA I'm looking for, and this is the kind of parameters, and this is the control strategy for how do I want to aerate the bioreactor with, say, air or oxygen or a combination of CO2 and nitrogen. So this is how the process parameters are decided for the manufacturing scale bioreactor. And what you would check for is, say, for example, if I'm projecting for certain gas flow rates to be used here, are the MFCs, which is the mass flow controller for the gases, are able to deliver that? Is the agitator able to even go to the RPM which I am proposing to achieve a certain power per unit volume? And is the bioreactor able to maintain the temperature well within a very short range, which is like plus or minus 0.5 degrees? Because cell lines and some of the components in the media, like vitamins, are thermolabile. So all this has to be kept in mind while scaling up a process. So when the process was scaled up to pilot, the lab scale is in blue color and the pilot scale is in maroon. So you could see that the cell counts were matching between the two, perfectly matching, and viability was also matching, and the titer output was found to be the same. So this gives you enough confidence of going ahead with the at scale process, right? So the same thing was done, but you are seeing the results for here. So the green color one is the manufacturing scale. So what we observed is that first of all, the cells were not able to get to the higher cell count, or the peak VCC was not the same. Viability started crashing early days, too early than the others. So this is a very bad situation to be in, because once the cells start crashing, they release all the dirt into the, or the garbage from within the cells to outside the broth. So in the broth, if you have proteases released into the solution, then you have product fragmentation. And also, because now you have a lower VCC and lower viability and the batch ended early, first of all, the productivity per day was not high, and the overall the titer was less than half. So these are a nightmare for any upstream person, that you have a perfectly matching pilot scale data, but your manufacturing scale data is not matching. So we started with something called as a RCA. RCA stands for the root cause analysis. So root cause analysis is you can probably Google this, but there is lot of ways you can solve this. So fishbone diagram is something which is commonly used, or 5 whys. You can use this kind of analysis also for your personal life. So uh, there were three major areas which were focused. One was about the scale-up strategy, and another was inoculum generation. So when I talk about scale-up strategy, right, this is like, have we decided the right parameters? Was the DO percentage the same? Was the pH point, set point, and the levels at which it was maintained the same between lab, pilot, and manufacturing scale? Similarly, the DO gassing strategy, whether the cells were seeded at the same density or not. So all this is the scale-up strategy. Next is the inoculum generation, which is like, was the vial thawed properly? Was the operations fine where the cells are not kept out for more time so that they starve themselves? And others would be that media and feeds, because at the smaller scale, you prepare media at 1 liter or say a few hundred liters. But once you go to a larger scale, you're supposed to be preparing these in thousands of liters. So could anything would have gone wrong in preparation of media or feeds? Was there something which was missed out? So all this could be probable causes. And what was done is, because this was the first scale up, right? So there is something called as a satellite run. So what you could always do is, once you have inoculated the production bioreactor at the manufacturing scale, you could take a sample out of it and also run it in a flask or a 1 liter bioreactor, right? Which could serve as investigation aid for you. So that was done for this first scale up batch. So here, what it does is that whenever you're running a satellite run for the production bioreactor, it captures all the variability coming in from media preparation to vial thaw, the inoculum, all of this prior to the production bioreactor is captured. So say if your scale up, okay, so if the, okay, we'll go one by one here. So you can start at different stages. For example, if you're capturing production bioreactor, then what could happen is that you'll capture all the variability here, but you'll not be able to pinpoint. So there it makes sense that you take a batch with the media prepared from a manufacturing scale. Next is that you could take a batch from the seed bioreactor, which is prior to the production bioreactor. So what you're doing is, you're first of all, with this experiment, you're nullifying anything which could have been caused due to the media and feed preparation. And with this satellite run, you could nullify, or you can negate any say root causes which are coming in from the vial thaw all the way till the seed bioreactors. So what could be seen here is that the manufacturing media and the R&D control, that ran well, but the manufacturing inoculum which was taken from the seed bioreactor, that was performing poorly. So what this tells us is that the problem was neither here, neither here. So it is something to do with around these seed stages. So this gives you enough confidence saying that at least, okay, the media preparations were fine, the way the production bioreactor run was fine. It's only that now we have a focus area, which is the seed bioreactor. So what was done is that the seed train investigations, again, the R&D, you are able to see this, right? That the shake flask to seed bioreactors, there seems to be something which is causing the seed bioreactor to not perform as well. So okay, so what was done is that the manufacturing inoculum was taken from the seed bioreactor as well as the inoculum stage, that is the 2 liter shake flask, and then those kind of match the trend seen for the production bioreactor, the one which is gray in background, that was there. But whereas the R&D control was fine using the same media, and there was no viability crash. So here, what could be clearly seen is that the problem seems to be not even the seed bioreactor; it is emanating from the shake flask stage itself. Therefore, ruling out any production bioreactor issues or the seed bioreactor issues. So now we have come to a conclusion saying that the media was not an issue, and neither was the satellite batch inoculum generated manufacturing was the issue. Or okay, so the problem what we were able to locate is that it is in fact the shake flask inoculum which was causing the issue. And shake flasks are usually kept in a shaker incubator where they are incubated for certain days. Usually this lasts for about 6 days all the way till sometimes even 15 days. So the failure of the inoculum at large scale usually is that for if I have to put it this way, that for inoculating a 1 liter, you'd require roughly around 100 ml. But whereas for inoculating a 2000 liter bioreactor or a higher scale bioreactor, you'd require inoculum close to 200 liters. So there is a difference in the scale, right? Scale I meant is from 100 ml all the way till 200 liters. So for generating this kind of inoculum, you would require to do sequential passages, thereby expanding the volume with each passage, which is 10% inoculum goes into the 90% media, and then the same is continued throughout. So this causes the cells to age. So just the way we humans have generations, right? That you have grandparents, your parents, and you. So you are three generations. So cells also have a generation number. So more is the generation. Usually for the recombinant cell lines, we have issues with cells undergoing stress or genetic changes or mutations, which could lead to growth issues. So what was determined is that we had this as one of the root causes, saying that this could be extended cell generation or seed bioreactor operations or production bioreactor operations. Now, when the inoculum was seen, it was normal in growth rate and doubling time, but it failed at production run. So when we were discussing about the shake flask, right, and the shake flask being kept in incubator, so the heating of the bottom of the flask was found to be the underlying reason, because the 2 liter was found to be the root cause. And then on further investigation from temperature mapping, it was found that the bottom of the flask was getting unusually heated, thereby leading to a temperature of more than what it was supposed to be. So for example, even if you run a culture at 38°, you will see these kind of effects if the set point is 37. That is why you have a very tight range for all the cell culture operations. So this is something which is the least expected stage, right? Because if you really see, the flask is not the stage which is the scale up, but these kind of things do happen when you go from one facility to another facility. So, and also what it did point out is that whenever you run a satellite run, that is the control runs to the production bioreactor, it always helps you in arriving at the root cause. So whenever that just tells you that how important are the controls to any experiments you do. So what we saw was that after resolving the issue associated with the bottom plate heating for the flask, even then what we could see is that the batches which were taken later, the cell culture performance was better than for the first batch. However, the performance was not the same as the small scale batches. So again, the similar root cause investigation was done to identify the suboptimal performance. So you're able to see here that instead of crashing here, the cells did reach the peak cell count, but however they crashed on a few days early, and also the viability was maintaining better than the first batch, but they did crash later. Also titer was slightly better, but there were still issues persisting. So what did we miss this time? Like, we had seen the scale up strategy. Now we have seen the entire inoculum generation. We had seen the media and feeds, but even then, why is the cell culture not performing? So now the focus shifts from the shake flask to the production bioreactor, right? So what we found out is that one of the feeds which was used for the commercial process, then here we had it was a multi-component feed, and one of the components were missing from the feed, and this is coming in from a very reputed vendor, with whom obviously I cannot share the name for for. But this did happen where they gave a certificate of analysis saying that, okay, this feeds meet your requirement, and these are certified to be certified from our end. But however, when we did the investigation, we found that there was an issue. So you could clearly see here that this commercial feed, which was there procured at R&D, was yellow in color. The one at manufacturing was almost colorless. And this was an in-house preparation of the same feed, which also had a certain color to it. So when you have something called an osmolality measurement, which is nothing but the depression of the freezing point caused by the solutes present in the solution. So here, the osmolality was in few hundreds, but for the colorless solution, the osmolality was only 18 millimolar. And also, when the glucose was measured, it was found that the glucose was higher in this, but here it was lower and here it was nil. At the same time, when the pH was looked upon, this was something which was very stark because the pH here are in the range of 7. However, the pH for the commercial feed procured was 3.6, which is really shocking, right? First of all, it is supposed to have some color to it, doesn't. Osmolality is supposed to be a representation of the amount of solute present in the solution. It is clearly that the required amount was not present, therefore the osmolality of the solution was much lower. And also because the required solutes were not present, the pH of the solution was way lower. So why was it not identified? Here in vials, it's clearly visible, right? The problem was that it came on large pack sizes where you have this translucent bags and you cannot clearly make sure that the colors are matching. It is something which is visual and it was scaled up for the first time to manufacturing. So this is one of the easier to miss. But however, once you alicode this and when we were able to alicode this into vials and see this, we could see that there is obvious differences which were there. So these are the chromatographic evidence. So obviously you would like to find out what the components were, right? So it was subjected to a RP-HPLC, and what was found is that you're able to see the peak intensity here, right? This is milliampere units. So here the peak intensity goes all the way till 1500. Here the peak intensity was not there at all. There was very minimal components which were there at a certain, the same peaks were present, but not at the same intensity as required. So you could see that the maximum peak you see here is roughly around 25 MAU, whereas here you have 1500 MAU. So what was identified is that the concentration of the commercial feed produced at manufacturing was not identical. And also there was a comparison of the total gas Paget flow rates done, and it was observed that for manufacturing we are using higher vessel volumes per minute than the pilot scale. So what was done is that we decided to switch to a microsphager. Usually in mammalian cell culture, whenever you're scaling up, the convention is to go ahead with a macrosphager. But what we observed is that because we ended up using too much of macrosphager, some of the metabolite profiles were not fine. So this is where we decided to change to microsphager and reduce any kind of further risks. So these were the old profiles which we had seen, and the green one is with all the issues fixed. So here you could clearly see that you are able to achieve the cell counts a bit earlier itself because might be of better conditions, and also the titer, the viability, everything was matching with the small scale and the pilot scale. So a few things which helped is one is changing the commercial raw material, fixing the shake flask issue, and also fixing the aeration strategy. So this is something which helped in having a successful campaign of the scale up. So whenever you're scaling up, do not leave behind any residual uncertainties, as in don't assume anything. So you should have everything jotted down. You have to do a proper risk assessment for scale up and thereby remove any kind of uncertainties which could be present. If you have uncertainty, it's always better to have this sorted out before so before any scale up activity is taken. and also cumulative effort. That is, all the groups have to come together, right? So parallel investigations at R&D, the manufacturing, supply chain, and also the vendor. This is something which reduced the time significantly. Instead of spending months together, this was resolved within a matter of few days. And also, whenever you're working with a vendor, right, you don't have to blame the vendors always. So a lot of vendors, they are forthcoming, and they what they do is they also help you in fixing a lot of issues. And say, for example, if you're looking for certain solutions to your process, they are always open. So for example, if you're looking for a particular kind of filter for filtering the media or feeds, uh, they always are ready to help you because they have their own commercial interest, right? Because if it's successful for you, then obviously they get the business. So some of the challenges during the development was that in the smaller scale, right? As in, uh, there was a change in the inoculum propagation for the manufacturing scale. That is, because now we had to scale up the operations, we had to move from the fixed assets, which is the stainless steel, to disposables, and also there was a change in mixing from a STR. STR is nothing but a stir tank reactor to a weighbag type of a reactor. So here some of the stages were stir tank which were replaced with a weighbag. Now weighbag has a more gentler form of mixing. So there was a comparison which was done for the stainless steel and the disposables. And also because now we have gone ahead with a different mixing itself, because here the predominant form of mass transfer is through agitation and aeration. However, for the weighbag, the mass transfer is happening through the overlay. That is the headspace of the bioreactor rather than the sparging. So here, what was seen is that when we moved to the disposable inoculum, we could see that the cell counts were lower and also the viability was lower. So, and obviously when the cell count and viability is lower, because each cell contributes to the total titer, right? We could see that the total titer was lower. So again, so what it indicates is might be the inoculum stage was not optimized properly. So we had to go back to the inoculum stage and see how the KLA is looking like. Why did it fail to perform? Was there any temperature issue? So there were two things which was done. One is the mechanical operations of the wave bioreactor were looked into, and also the effect of individual seed train unit operations were also looked into. So here, what could be seen is that whenever the inoculum transfer was done, this plate which is rocking was the rocking was stopped during the inoculum transfer or sampling, and this happens for close to 30 minutes to 1 hour. So what was seen is that because the temperature set point was not removed, it was still in a temperature control loop. So what that means is that the temperature sensor is located elsewhere, which was in a colder region, and the culture was located here. Thereby, the heater was getting heated more than what it was required to maintain the temperature, and because of which the temperatures was reaching 39, and that was leading to lower cell counts and underperformance of the culture. So with the fixing of all this inoculum issues, you could see that the purple line is the final one. So here you could see that the VCC was better, the viability was better, and also you are able to get the same output as the stainless steel bioreactor, which is the green color line. So in summary, communication is the key. So who would have thought that stopping the weighbag during the inoculation could have come across? So this is through like regular training and monitoring of the operators, and also to educate them on, say, what are the issues which could be observed for scaling up of a process, or what are the things to watch out for. And also there was a lot of cross-functional support between manufacturing and R&D for getting this done. So it is always a pyramid of knowledge, right? So first you have to understand the process as much as possible. That is, you need to know what is that which can cause the cell culture to not perform. And once you have this, then you have to document all of this in documents which are like process characterization summary, or they say the document which tells you clearly that which are the parameters which could make the process swing from a success to a failure. And also cross-functional communication. So we have manufacturing, which is including the production shop floor and the MSAT and QA, QC, all this. So you need to have all the cross-functional communication. You should have the network. And also there is a new thing in the market, which is nothing but the PAT. This is other process analytical tools. So instead of finding out later that the cell counts have gone down through routine sampling, that is sampling usually done once a day, right? You have now biomass probes, the NIR probes, the Raman probes, which can tell you online as to how the parameters are looking. That is the how the glucose concentrations are looking, how the amino acids looking, and how is the waste metabolites, which is lactate and ammonia concentrations looking like. And you can always have an online comparison with your historical batches. So this kind of, and so we are on time at 3:15. So there should be another 15 minutes of Q&A.
Students, if you have any questions, please unmute yourself and ask. Any questions? Any question not
Salma?
related
Okay.
to subject?
Yeah, it could be any question. Like Manjunath sir said, higher education or what to focus on. That could also be a question, because we have another 15 minutes to spare. Right after this, I'm supposed to be moving into another meeting. Salma ma'am, the chat box is not on. Is it okay? It's only for the team members. Fine.
Yes.
Okay.
No questions, so we will then move towards concluding the session. I now request Ms. Ashwati to please propose the vote of thanks.
Good afternoon, everyone. Good afternoon, everyone. Honorable Chairman, Director, respected Principal, the Head of the Department, and my dear participants. It is my privilege to have been asked to propose a vote of thanks on this occasion. I, Ms. Ashwati Unnikrishnan, Assistant Professor, Department of Biotechnology, the Oxford College of Engineering, extend a very hearty vote of thanks to Mr. Abdul Wahid, Biocon Biologics India Limited, who is also the alumni of the Department of Biotechnology, batch of 2003, in joining us today and sharing your expertise in the field of upstream process development with us. I thank our chairman, Sri SNVL Narasimha Raju Garu, Dr. Amarnath K, Director, the Oxford Educational Institutions, Dr. Aravind AS, Principal, the Oxford College of Engineering, for the support. I am very grateful to Dr. B.K. Manjunath, the head of the department, the Oxford College of Engineering, for his constant encouragement and support. I thank Mr. Rohit B.H., Student Coordinator, Department of Biotechnology, for his involvement and willingness to make the webinar a great success. I thank my dear colleagues for their support. I also thank the participants for their active participation in the event and making it a great success. Thank you all. Thank you so much.
Thank you, Abdul. Thank you for being there. I